Hello, my name is Kieran Howard and I'm a PhD student in the School of Biosciences at Cardiff University, supervised by Nathan Harmston. As part of my project titled Understanding the Structure and Function of Conserved Non-Coding Elements Using Data-Driven Approaches, we have developed nf-CNE, a Nextflow pipeline for identifying conserved non-coding elements which I'm going to present and demonstrate for you today. Here is an overview of the poster and I'm going to go through it one section at a time. Conserved non-coding elements, or CNEs, are sections of DNA outside of protein coding regions that have been highly conserved across large evolutionary distances, indicating a strong purifying selection over tens or hundreds of millions of years. Here we can see a conserved non-coding element located near developmental gene IRX1, showing a high degree of conservation between multiple species. Several studies have shown that CNEs are not randomly distributed throughout the genome, instead clustering in the vicinity of genes encoding developmental regulators, pointing to a probable role in gene regulation, particularly in the regulation of key developmental genes. Despite their importance, identification of CNEs is non-trivial as, by definition, these elements do not code for proteins. While some databases of CNEs do exist, such as Ancora or UCNE base, they are limited in the species that they have analyzed and use preset filters that cannot be changed. Additionally, current tools for identifying CNEs, such as CNER or CNE Finder, can be difficult for novice users to use and offer limited customizability and modularity. Here we present nf-CNE, a Nextflow-based pipeline designed for the robust detection of CNEs between two or more species. This pipeline addresses the need for a user-customizable and scalable workflow for CNE identification using Nextflow's modular design and parallelization. It offers flexible configuration of each analysis step with user-specified parameters. It uses integrated filtering of whole genome alignments using any UCSC data track or tracks to ensure high confidence in the conserved elements detected. It accepts a set of reference and query genomes or pre-generated pairwise alignment files, accommodating comparisons between both closely related and highly divergent species. It generates a structured and standardized output format, for example bed coordinates of the CNEs, that can be used for downstream analysis, utilizing a many-to-many numbering system to track CNEs with multiple mappings between species. The primary input of nf-CNE is a tab-separated values file which contains the species names, the path to the reference genome for that species, although this is optional and it is downloaded if it's not provided, a path to pre-made net.axt files, this again is optional and will be generated if it's not provided, and can also be optionally gzip compressed or uncompressed, a comma-separated list of UCSC tracks to filter, and a comma-separated list of tool-based filters to apply, although currently only BLAT is supported. The TSV also contains several entries that are thresholds or options specific to these above tool-based filters. This is an overview of the workflow where we have our inputs at the top, and any steps that are not dependent on previous steps are run in parallel, and additionally several steps, such as the downloading of UCSC tracks, will launch multiple processes so that if you have, for example, 6 tracks to download, all 6 will be downloaded simultaneously. The primary output of nf-CNE are 2 bed files containing the identified CNEs, one with coordinates referring to the target species and the other with the coordinates of the query species. Each CNE has an associated number, for example CNE1, creating a many-to-many relationship where CNEs with the same number are homologous. Additionally, any downloaded UCSC tracks are kept that can be used in downstream processes. Here we can see the same CNE that was shown in Figure 1, identified between human HG38 and mouse MM10 using nf-CNE shown in IGV along with the UCSC FastCons 20ROI track, showing a high degree of conservation within this CNE. And in conclusion, nf-CNE delivers a flexible, containerized workflow for discovering conserved non-coding elements across any pair or set of genomes. Users may supply their own assemblies or rely on automatic downloads, and parameters can be tuned without any code changes. The resulting cross-species bed files have a many-to-many relationship, enabling identification of CNEs that are in multiple places within the genome. Because nf-CNE is packaged with containerized environments, every run is completely reproducible, with planned updates to include CNEs using multiple sequence alignments along with additional filters. nf-CNE lowers the barrier to exploring conserved elements between any set of organisms. Thank you for listening, and I'm now going to give a quick demonstration of running the workflow. On the right hand side of the screen we have our input TSV file showing that we are doing a comparison between HG38 and MM10. I've provided a path to the reference genome for both species and also provided a small cut down example of the pairwise alignment files used for testing and demonstrating. These are also available in the GitHub repository. The UCSC tracks we will be filtering out are GAP, the NCBI RefSeq curated list, and repeat masker for both species, although the tracks don't have to be the same between species. and we will be performing additional filtering using BLAT, removing any putative CNEs that have at least a 90% similarity to 4 or more regions within the genome. By default, nf-CNE will use the file input.tsv along with a columns value of 50 and an identity value of 50, though these can be changed with the input, columns, and identity options. If we run the workflow, which I will fast forward, we can see that the download reference and gunzip file processes were not run as we provided both reference files and our input pairwise alignment files were uncompressed. If we look in our results folder, we can then see that we have our final filtered bed files along with the UCSC tracks that we downloaded. Here we have a similar setup to before, however we have removed the MM10 reference genome and gzip compressed both of our input files. If we run the workflow again, we can see that this time the download reference and gunzip file processes were run, and additionally the reference genome is downloaded and in our results folder.